Breast cancer is the second most common cause of cancer-related deaths in women. It is responsible for 42,000 deaths annually in the United States. Breast cancer occurs when mutations induce abnormal growth of breast cells, forming a tumor that can metastasize throughout the body. While some risk factors are well known, such as the BRCA1 and BRCA2 genes, women with no known risk factors still develop breast cancer. Screening technologies such as mammograms aim to detect breast cancer in its early stages, but symptoms include a change in the appearance of the breast, such as thickening of tissue, dimpling, peeling, scaling, and flaking of skin. Breast cancer mortality rates have largely declined with the advent of hormone-based therapies, but there is still a significant health disparity between Black and white women, in part due to the biology of cancer. Black women are more likely to be diagnosed with triple-negative breast cancer, a subset of breast cancer that lacks a receptor status that makes hormonal therapy an effective treatment option. Mutations that cause breast cancer often interfere with the cell cycle. The cell cycle is a process in which a parent cell grows and divides into two daughter cells. Occurring in 4 stages, this process is essential to maintain the structure of a multicellular organism. While cell division is a ubiquitous process, inappropriate cell division can cause diseases such as cancer. There are many mechanisms that can proliferate the growth of breast cancer, one being the epidermal growth factor receptor or EGFR signal transduction pathway. In the normal EGFR signal transduction pathway, an extracellular signaling molecule leads to cellular division. The signaling pathway begins at the EGF receptor. This receptor consists of an EGF binding site on the extracellular domain and tyrosine kinases on the intracellular domain. The EGF receptor has 2 identical subunits that are normally dissociated. First, epidermal growth factor, also known as EGF, binds to the EGF receptor. When EGF binds to the extracellular domain, a conformational change induces dimerization of the protein subunits. The new conformation allows for cross-phosphorylation of the intracellular tyrosine kinases. Once phosphorylated, the EGF receptor recruits GRB2, which recruits another protein called SOS. SOS binds to a G protein called RAS, which is bound to GDP. Once SOS and RAS are associated, the GDP is exchanged for GTP, activating the RAS protein. Still associated with the membrane by a lipid domain, RAS travels and activates the protein kinase RAF. RAF activates other protein kinases called MEKs, and MEKs activate ERKs to continue the signaling cascade. Activated ERK moves into the nucleus and activates MIK, a transcription factor. Activated MIK accumulates in the nucleus, dimerizing with MAX. The MIK-MAX dimer can bind to specific DNA motifs to induce transcription of cell cycle genes. MIK-MAX can also bind to histone acetyltransferases to acetylate and open the structure of DNA and promote transcription of cell cycle genes. The EGFR signal transduction pathway is normally regulated with phosphatases. Phosphatases remove phosphate groups to prevent the signal from proliferating uncontrollably. There is also a mechanism to turn off the RAS-G protein. RAS has GTPase activity, meaning it can hydrolyze GTP to GDP and turn itself off. While the EGF signaling pathway is important to healthy cell growth and division, it can also play a role in cancer growth and proliferation. The EGFR gene is located on chromosome 7 and consists of 28 exons. Mutations, such as deletions in exon 19 and point mutations in exon 21 in the tyrosine kinase domain, contribute to EGFR amplification, causing the signal to proliferate without EGF binding. Gene amplification can also result in an overabundance of the EGFR protein. This can lead to ligand-independent phosphorylation, meaning the signaling pathway is activated without EGF binding to the extracellular domain. Lapatinib is one drug that inhibits the EGFR pathway. It works as a tyrosine kinase inhibitor. By binding to the intracellular phosphorylation domain, it prevents autophosphorylation from occurring when a ligand binds to the extracellular domain. This inhibits the resulting pathway that leads to cancer proliferation. Cetuximab is another drug that is used to treat EGFR-positive triple-negative breast cancer. Cetuximab is a recombinant IgG1 monoclonal antibody. Through competitive inhibition, it prevents epidermal growth factor from binding to the extracellular domain of EGFR. As a result, the signaling pathway does not propagate. While the EGFR signaling pathway plays an important role in cell division, mutations in the EGFR can contribute to diseases like breast cancer. As researchers increasingly understand how this pathway contributes to disease, there is potential for new therapies and the possibility of addressing continued health disparities.